Hello students, this is Dr. Faizan Mirza. We are discussing protein mediated transport, facilitated diffusion and active transport. So what's facilitated diffusion? Diffusion, as you know, it's occurring down the gradient. Facilitated diffusion basically is a type of diffusion where a mediated transport is involved. The mediated transport means that here a protein, which is a channel protein or a carrier protein, will be involved in moving the substance down the gradient. This type of transport is not linked with metabolic energy. It is involved in equilibrating the solute concentration in ECF and ICF. And since it is not linked with metabolic energy, which means ATP breakdown is not required, so any respiratory inhibitor that can cause ATP to deplete in the cells will not affect facilitated diffusion. For example, this phospholipid bilayer I am drawing to you, these green dots here, they represent a solute that must enter from ECF to ICF, and this is present right outside the cell membrane, and this is the carrier protein which is to accommodate moving this substance and mediate its transport into the ICF. For the carrier protein to allow the movement of this substance into the ICF, this transport protein, since it's not a channel protein, there is no pore here, this carrier protein must bring about certain conformational changes. So the binding of the substance or the solute at the ECF surface, it triggers a conformation change. Conformational change means the change in shape of the carrier protein. Once the shape changes, the substance is released from the ECF into the ICF, and this is how a carrier transport protein moves the substance in one direction. Energy for conformational change of this transport protein is not linked with breakdown of ATP. It is just coming from the binding of the substrate or the substance moving from the ECF to the ICF. The solute or the substance must be coming to a specific targeted protein, because only the specific solute will be allowed to enter or leave the cell through the carrier protein, because every solute will have its own carrier protein. So the transport proteins are very specific, and conformational change will only be triggered if the complementary solute or the solute which is complementary to the transport protein is actually there. What is active transport? Active transport is also a protein mediated transport, but here the energy of conformational change could either be coming from the breakdown of ATP, in which case it will be primary active transport, or the conformational change will come because of the energy which is stored in the electrochemical gradient, in which case it will be a secondary active transport. Since this is using metabolic energy, so any inhibitor that can deplete the ATP reserves in the cells or prevent ATP synthesis in the cell, it will affect this active transport, and it will actually cease down. I'm drawing this phospholipid bilayer here, and this is a cell membrane. This phospholipid bilayer, you can see it will have a surface facing ECF, another surface facing ICF. This is a sodium potassium pump. In sodium potassium pump, there are, again, as it's a transmembrane protein, it's a carrier protein, so the ICF surface is having a binding site for 3 sodium ions, whereas the ECF surface is having the binding site for 2 potassium ions. When the 2 potassium ions are bound to this particular end at ECF, and 3 sodium ions have occupied their slots on ICF surface, the enzyme ATPase, which is present here at the ICF surface on the same protein, cause ATP to break down into ADP and PI. The breakdown of ATP into ADP and PI releases energy, and that energy is used to bring about the conformational change. So the conformational change which occurs on sodium potassium pump is because of 3 factors: 3 sodiums attached at ICF, 2 potassiums attached at ECF, and ATP being broken down to ADP and PI. All these 3 things must occur simultaneously for this sodium potassium pump to function. This caused 3 sodium ions to leave the cell, and 2 potassium ions, they move into the cell, and this is active transport. So both the ions are moving against the concentration gradient. When 3 positive charges, which are sodium, they are leaving the cell, they move out, and 2 positive charges are entering the cell in exchange of 3 sodium ions going out. It means that a net deficit of 1 positive charge will start to build up inside the cell. This causes an imbalance to occur. And this imbalance is reflected in the cell membrane gaining a negative charge along its interior, where a net deficit of one positive charge kept occurring because one excess positive charge kept going out in exchange of two positive charges coming in, because three sodiums left while two potassiums entered. The 3:2 ratio actually causes imbalance. So outside the cell there is a high positivity, inside the cell there is a negativity, a relative negativity though. So this is why the cell membrane, once it gains this potential of imbalance of charges, this is primarily because of sodium potassium pump. That's why the sodium potassium pump is also called as an electrogenic pump. How active transport is occurring in sodium potassium pump, how it works, I've already discussed with you. The sodium potassium ATPase, it works in such a way that because of this, the sodium concentration outside the cell is 10 folds higher as compared to inside. There are subunits, alpha subunit and the beta subunit. The alpha subunit is having about 100,000 Daltons of molecular weight, whereas the beta subunit is 50,000 Daltons. The catalytic activity occurs on the alpha subunit. The sodium potassium pump pushes 3 sodiums out and 2 potassiums in, and ATP is broken down to ADP and PI. Already covered that this is an electrogenic pump. So when 3 sodiums go out and 2 potassiums enter in exchange, a net deficit of one positive charge keeps building inside the cell. And this is because of this excess of positive charges going out, or the lack of equal positive charges coming in, that the cell starts to become relatively negative along its inside. And that's why we call this sodium potassium pump to be an electrogenic pump. This negativity inside the cell is the relative negativity. The cell membrane is relatively negative when compared to outside. So you actually place one electrode inside the cell and you place one electrode at the surface of the cell. So the difference of the two electrodes, the surface electrode and the one which is placed inside, is actually deflected, or you can say is reflected in the deflection of the needle or the voltmeter reading, which can be placed here, and it tells you the exact millivolts of how much charge is present inside, which is negative, as compared to the one which is outside. Secondary active transport. Secondary active transport, as I already told you, makes use of energy stored in the electrochemical gradient. So here we'll discuss co-transport, which is symport mechanism. I'm drawing epithelial cells. These are having microvilli at the surface. This is the tubular fluid, and these cells belong to the proximal convoluted tubule. These proximal convoluted tubule cells are present adjacent to a blood capillary. You know this blood capillary will have red blood cells and plasma. This is the microvilli region. This is called as the apical surface, which faces the tubular fluid. So whatever filtrate is there in the nephron, which eventually will form your urine, will be present here. And the apical surface will be in contact with that fluid. The opposite end of the same cell will have a basolateral surface. This basolateral surface is the one which is facing the blood capillary. The blood capillary, you know, is having a single endothelial lining as well. There are transport proteins here which are co-transporters, sodium and glucose co-transporter, which allows sodium and glucose to enter together inside the cell. Once sodium and glucose are they enter inside the cell, they can then move into the blood by diffusion. Similarly, you have sodium and amino acid co-transporters present in the same membrane, and again sodium and amino acid can get absorbed. So this is how selective reabsorption of these solutes it occur in your kidney tubules using secondary active transport. Here the energy from ATP is not directly used. The energy was stored in the electrochemical gradient. The same process occurs in our intestine as well. In the villi cells, we also have the same channels here, the same carrier proteins here, which ends up reabsorbing the solutes or these nutrients from our duodenum into the blood. So this depicts the reabsorption of nutrients into the villi in the duodenum as well. So how was this occurring? So this is the phospholipid bilayer of the cell. And the phospholipid bilayer of the cell again will have one side which is facing ECF, the other side will be facing ICF. So this is how the protein would be. It would be allowing sodium and glucose to move into the cell, and you can see that this is not a channel protein, it's a carrier protein. The pathway is not running, the pore is not like continuous here. So the ions, the sodium ion and glucose, they enter together and they move into the cell by the process of protein mediated transport. Counter transport occurs when two substances are being moved in opposite direction. For example, antiport. This is also called the antiport movement. For example, sodium amino acid exchanger, where sodium leaves the cell and amino acid enters the cell. What's an electrochemical gradient? The electrochemical gradient can, if this is a chemical gradient. There is a high concentration of glucose inside and a low concentration outside. So this is a chemical gradient. So you know that glucose is likely to diffuse out or move out because if given a chance, because there's a diffusion gradient here. So this is an example of a chemical gradient. But for example, if I discuss the gradient of a charged substance having a polar structure H plus and H plus high inside and low outside, so what do you see? We see that this gradient is not just chemical gradient, it is electrical as well, because it causes the distribution of charges, because when there is a lot of H plus inside, there will be highly positive charges inside, and outside the cell will be a bit of negative as compared to inside. So this is called as an electrochemical gradient. For instance, if you start to just again apply the phospholipid bilayer and try to picture it, how the exchangers are working. So the amino acid and sodium exchanger will be moving sodium and amino acid in opposite direction at the same time, which means that the amino acid will bind to the surface at the ECF at the carrier protein, and sodium will attach to the surface at ICF of the same carrier protein. When these binding sites are occupied at the same time, conformational change will occur in the protein, and sodium will be released outside and amino acid will be released inside, and the carrier protein will then again turn back into the same position in which it was initially. That's all I'll be covering in this video. Thank you so much.